When we started talking about the purpose of the scientific method, we said that we need this in order to objectively determine how the world works. And we kind of alluded to the fact that there were some built-in safety mechanisms to prevent things from leading us to incorrect conclusions. Basically, what we're going to talk about in this video and in the follow-up video is some of the features that are in the scientific method that help prevent us from making inaccurate conclusions. The four that we're going to really focus on in this class is that the studies need to be unbiased, they need to be as controlled as possible, and these are the two features that are going to be talked about in this video. They need to be repeatable, and they need to be ethical, which are the two factors that we're going to cover in the follow-up video. So let's just kind of go through this list chronologically, starting with unbiased. Bias is definitely a word that's kind of used in everyday settings, but what would that look like specifically in science? A bias is going to be when something other than reality or the truth influences the conclusion of our experiment. And it doesn't matter whether it's intentional or unintentional. Either way, it would be considered a bias. The issue with the bias is that if it's forming our conclusion, then our conclusion likely is inaccurate, or it's not a true reflection of how the world actually works. It's a reflection of that bias. There's lots of different kinds of biases that could theoretically be problematic, but there's kind of three biggies that we tend to see in a lot of scientific experiments that we really want to take care to avoid. The first is something called sample bias, and it has to do with how you sample subjects. So if you think about any given variable that's being tested in science, you're usually talking about how this variable affects a very large demographic of individuals. For instance, if we go back to my whole initial scenario where we talked about how temperature might affect the ability of chickens to lay eggs, that statement is kind of referring to chickens very generally. There's a lot of chickens in the world, and there is no way we could capture all of them and test all of them to see if they all react the same way. So because that's not possible, what we do in science is we do something called sampling. We will take a subset of individuals that we actually apply the treatment to, and we call those our subjects. And the idea is those subjects will reflect how all individuals would do, which we call the population. So the population is all units that our research question would apply to. And then the sample is the small subset that we actually include in our study as subjects, okay? So what sample bias is talking about is it's referring to the sampling process. Sample bias is when our sample when they don't reflect the population as a whole. If the sample does not represent the population well, then how they respond does not necessarily give us good insight as to how individuals in the entire population would respond. So in order for us to be confident that the population as a whole will respond the way that our sample subjects are responding, we need to make sure that the sample is a proper reflection of the entire population. If it's not, that is when we would say that sample bias has occurred. Confirmation bias. Confirmation bias is when you either seek or, sorry, that's an or instead of, or interpret information so that the conclusion matches your initial thoughts. Maybe another way to state this would be like seeking information that shows that you are right, okay? So you're trying to confirm your initial beliefs, and that is influencing how you either interpret the information you have, or you are specifically choosing some information and excluding other information that does not fit your original beliefs. And then finally, the placebo effect. The placebo effect is a psychological phenomenon. So this is mainly going to be an issue where humans are involved. Where subjects either report or experience improvement, and it could be improvement in whatever is being measured, because they expect a treatment will help them. So basically, these subjects are either reporting improvement, or sometimes we can actually physically measure changes in them physiologically. That is an improvement because there is an expectation that treatment of some type will be beneficial, not because the treatment actually is providing any actual significant improvement. So obviously these biases are problematic. We don't want them to influence our outcome. So what can we do to avoid them? Well, in terms of sample bias, usually the way that we prevent this is, I guess, twofold. We need to make sure that we identify appropriate sample pools. A sample pool is going to be, essentially a list of subjects or a group of subjects that you can pull from. You don't necessarily test everybody within it, but you kind of identify, this is where I could recruit some subjects from. And then once you've identified those sample pools, kind of the other main way that you're going to minimize sample bias is by random sampling. We want to randomly select the individuals from these sample pools so that we don't either intentionally or unintentionally cherry pick individuals that might lead to an outcome but might not reflect the population as a whole. Kind of the thought is that if we sample randomly, then we'll have a greater likelihood of it reflecting naturally the true population. In terms of the placebo effect and confirmation bias, well, the way that we're going to minimize these is with blind studies. And there's actually two different kinds of blind studies that can occur. The first type of blind study is called a single blind study. In a single blind study, the subject doesn't know if they are receiving the true treatment. Essentially, in a single blind study, the subject doesn't know what treatment group they've been assigned to, okay? Now you might be asking yourself, how is that possible? Like, for instance, if I'm testing a medication for blood pressure, aren't the subjects gonna realize if I don't give them a pill? Well, the way that we're able to complete these single blind studies and keep subjects in the dark is through the use of something called a placebo. A placebo is a treatment lookalike. It's something that mimics the appearance of the actual treatment, but it doesn't have the active ingredient. So it's a way of kind of making it so that the subjects don't really know whether they're getting the true treatment or not. Okay. So in the single-blind study, the subjects are not aware of whether they're receiving the true treatment. Okay. In a double-blind study, neither the subject nor the researcher interacting with them So neither the subject nor the researcher interacting with the subjects know which treatment each subject is getting. And occasionally, we don't want the person who is recording responses from the subjects to influence them in any way. So we might have a double-blind study where a third party is aware of who is getting what, but everything is labeled in such a way that the people administering the treatment and measuring the responses don't know whether the subject that they've just interacted with has the true treatment or not. And these are the two primary ways that we help minimize the effect of the placebo effect and confirmation bias. We also want to make sure that our studies are as controlled as possible, especially if they're a controlled experiment. And remember, controlled in the sense here means that things are being held constant amongst subjects. Constants are really important within controlled experiments in kind of two cases. One, we need to have constants between treatment groups. So for example, if you're testing a medication for weight loss, then you might give one group a placebo, you might give one group the active medication you're testing, but you would want to keep their diet constant. Diet is something that is definitely linked to changes in weight. So if you really want to know if the medication is having an effect, you're going to want to keep the diet the same. And that right there illustrates the why. We cannot determine cause and effect if more than one variable changes. So we need to keep those other variables constant in order to make the cause and effect conclusion at the end of a controlled experiment. We're also going to want to try and keep things controlled within the treatment groups as well amongst the subjects. So for example, we'd also want to make sure that not only is diet consistent between treatment groups, but we'd also want diet consistent within the treatment group as well. The why isn't necessarily cause and effect. It has to do with variation. When you are failing to control a variable, your variation of responses is going to go way up, and it's going to make it very difficult to determine if there are actually differences between treatment groups. So we could say a lack of controlled variables equals a lot of variation in responses. Some people might lose a lot of weight, some people might lose a little bit of weight, and if you have a lot of variation in responses, it's going to be difficult to determine the effect of the treatment. Because the responses will be all over the board. Now, kind of one thing to think about is like, we can't control everything, right? I mean, Subjects, especially living organisms, are really dynamic, and so it's impossible to keep every single minute thing exactly the same. So what do you do? What do you put your energy towards trying to keep constant? The things that we really want to mainly focus on keeping constant are factors known to influence the dependent variable. So for instance, if we're thinking about a medication that is going to change weight loss, we're going to be measuring the subject's weight. So we're really going to want to work on controlling factors that we know are linked to weight, like diet. Factors that we don't believe to have any influence probably won't be as critical to keep controlled. So, for instance, somebody's eye color. I have never seen anything published that would suggest that eye color is linked to weight in any way. So trying to keep that consistent, wouldn't necessarily be as critical as something that we know to have an effect on weight. That being said, there's going to then be those things that you just can't keep controlled. So what do you do to keep those from influencing your conclusion? Usually the way that we take care of all of those other tiny things that we just couldn't possibly look into all of them is randomly assigning subjects to the different treatment groups. Once we decide who our subjects are going to be, they are randomly placed into the treatment groups that are available, and the random assignment is supposed to prevent the accumulation of noticeable differences between subjects, between groups.